as Rob said, I'm based up at the Maroochy Research Station. If you've not been there, you should definitely come and have a look. It's a fantastic part of the world. People that drive in there say, oh, this is not a research station, this is like a park, and it is really well maintained. And I must admit that I never like getting in the car and driving the 100 kilometres down to Brisbane. I'd much rather be up there. But come by and see us anytime. You're always very welcome. I just wanted to start off by talking about the breeding team. Very important in any project, the team that works in it. And we've got Dr. Jodie Neal, who's our breeder in the team based at Nambour. Dr. Craig Hardner, who many of you would know, our quantitative geneticist. Dr. Alison Kelly from Toowoomba, the statistician. Mr. Paul O'Hare, who's the extension officer at Nambour. And that's the point I just want to emphasise, is that how important when you're working with an industry-funded project that you do have an extension officer involved with the project. And I'll explain a little bit about what Paul's done in the project later on. Our technical assistants are Eddie, Hal, Dougal Russell and Rod Daly at Nambour and up at Bundaberg. And then in pathology we have Andre Drenth and Femi working on the pathology aspects of the project. Andres Killian from DART down in Canberra has done work for us with genomics. Ruth and Craig from New South Wales DPI are the entomologists. And then we've had Rob Henry and Kathy Knox collaborating with some genomics work. And I also do work with Jim Hannon and John Wilkie's small tree projects, and I'll talk just a little bit about the rootstock work there. When I was thinking about coming down and talking today, I thought I'll be selfish and I'll try and make it the talk structured to get the points I want to get across. And one of the things I do want to get across is that Dr. Jodie Neal, who's been a great breeder on our project, so good that DAF, who she works for, have said, no, we need her in the strawberry breeding project. So I'm losing her this year. She's been working on the project for about 4 years, and I'm losing her over this next 6 months. She's going to transition across to the strawberry breeding project. And so we're going to be advertising in the macadamia breeding project for a new breeder. So anybody that knows of potential candidates, please let them know, let me know. We'll be seeking a replacement over the next couple of months for Jodie as a breeder in the macadamia and also working on the peach breeding project. And the other part that I've got highlighted in red down the bottom is that we've got funding in this new macadamia project. We've just finished a 5-year project funded by Horticulture Innovation Australia. And this year we're just starting a new 5-year project, $2 million project, and we've got funding in that project for a PhD student. So we'll be advertising that in about October, and I'll talk a little bit more about that, and so please keep that in mind also. Okay, so to start off, what's the common link between macadamias and Australian rules football? They're both indigenous, that's true. Some people say you have to be nuts to be involved with either one.
Hawks protected species.
Yeah. Thanks, Chris. But another common link between the two things, between Australian rules football and macadamias, is this man. He was born in Scotland in 1827. He studied chemistry and medicine at Edinburgh and Glasgow. He moved to Australia, and in 1855 he was a teacher at Scotch College down in Melbourne. And in 1858, the very first game of Australian rules football was played between Scotch College and Melbourne Grammar. And this man was roped in to be the umpire at that first game. So that's his connection with football. He was the very first umpire of Australian rules football. And his connection with macadamia is that his name was John Macadam. And John Macadam, he was secretary and vice president of the Philosophical Society, or the Royal Society of Victoria. And on that society was another fellow by the name of Baron Sir Ferdinand von Mueller, who was the director of the Melbourne Botanic Gardens. And as these things happen, Baron von Mueller knew John Macadam, and when it came time to botanically classify this new plant, the macadamia, he named it after his mate, Macadam, and so we have the macadamia. So that's the link between, he's the link between Australian football and macadamia. I will just go completely off track here a bit more and say that Macadam, he died when he was 38 years old, so he died quite young. And in that time, not only did he have the Macadamia named after him and get involved with the football, he was also a parliamentarian. He was a member of the Legislative Assembly for Castlemaine. He was a lecturer at the School of Medicine at Melbourne University. He was also the Postmaster General of Victoria. All this, and he died at sea when he was travelling between Australia and New Zealand to act as a forensic pathologist at a court case over in New Zealand. So that's what he did in 38 years. Makes me feel pretty woeful at times. Okay, I'm going to start off by talking about macadamia breeding, and then I'll talk a little bit about peach breeding to finish up. So I'll give you some background about the industry, some results from our breeding project, and then talk about our future research. Australia is the second largest producer of macadamias. We were number one, but South Africa has pipped us. That may change again. 28% of the world supply, 6 million trees, 17,000 hectares. And I'm trying to think the other day. Someone said that the biggest horticultural crop in Australia is banana, and it is in terms of dollar value. But in terms of acreage, macadamia is actually larger because the trees are so big that it takes a lot of land to grow them. 40,000 tonnes produced annually, and 63% of the crop is exported. So when you're thinking about breeding, it's interesting to look at how many generations of work has already been done. And if you look at a crop like peach, the cultivar UF Sun, which is one grown commonly, it's 8 generations from the original cross of the temperate Southland peach with some wild, low-chill Hawaiian material. And if you go back with the temperate breeding, you could add another 8 generations onto it. If you contrast that with macadamia and one of the cultivars, a thing called cow from Hawaii, 344, which is grown in Australia now, it's 2 generations from the wild. So that's a huge difference in the 2 crops. What it means is there's great opportunities for breeding a macadamia. There's very little genetic improvement been done so far. The centre of origin for macadamia is here, right here on our doorstep. And so all the wild germplasm is just sitting out there in our backyard. And what this breeding project is about is producing new cultivars to make the Australian industry more competitive globally. It's not all roses. There are difficulties, and some of them I've listed just here. They're very large trees, 12 to 15 metres high, which means you can only grow 300 trees per hectare. The lifespan of the trees is 30 to 50 years, and so farmers don't change their cultivars too often. The nuts are harvested over 6 months, which makes it expensive, and the mature tree yield is poorly correlated to the young tree yield. This is some work that Craig Hardner did when he was with CSIRO, and he was showing the correlation between the different ages of the trees. This is the genetic correlations and the cumulative nut and shell yield. And you can see that the correlation with age 10 gets to 0.91 for the age 7 trees. And so you really need to grow the trees for 7 or 8 years before you get a good correlation with the mature tree yield. I mentioned before about the extension officer and how important it is to have an extension person in a project that's related to industry. Paul O'Hare organised that we consulted widely with industry to find out what their priorities were in terms of what should be incorporated into new cultivars. The number one priority by far was yield, and that was followed by these others. Husk spot, which is what Femi works on a lot, fungal disease in macadamia. Kernel quality was important, fruit spotting bug, and reduced tree size rounded it out. The way the breeding project has run is that we have a stage 1 where all the seedlings are grown, and then that's a single tree of each seedling, and then the elite seedlings are selected out and grown in a grafted, replicated trial, stage 2 trial. So the CSIRO program started in 1994 with cross-pollinations, produced about 2,000 seedlings, and they were planted, and the best 20 were selected in 2006. They were propagated by grafting to a regional variety trial. It was planted in 2008. It consists of 9 different trials on 2 rootstocks with 6 reps of each new elite selection. So it's taken a long time. It started in '94, and the good news for industry is that in 2 years' time, the first new cultivars will be released to them. So that's something they're really looking forward to. Obviously, we need to think very seriously about improving the efficiency of a program like that takes such a long time to produce new cultivars. And we've looked at a number of different methods. We've looked at a 2-stage tandem selection method that we've shown on a computer generated models that's more efficient, and we've got that planted in the field to actually see in practice whether that's true. We've done a little bit of work with Andres Killian on genomic selection, and that's looking promising. If we can cut down that stage 1 selection, that would be great. And then we've done some visual yield estimation on the seedlings to see if we can improve the efficiency of collecting data by visual evaluation of yield rather than actually hand harvesting all the seedlings. The way we do our controlled pollinations is that we bag all the racemes prior to anthesis so that we don't have stray insect pollination. Then we use that tube, as in the top photograph here, to put on the pollen that we desire, and then we rebag after that until the nuts have set. So very time consuming. That results in those seedlings that we just talked about, and they've been planted at a number of different trials. We've got, altogether in the breeding project, there's been 4 populations created, planted between 1997 and 2014, and close to 6,000 seedlings altogether, planted at 11 locations from Alstonville down in the south up to Baffle Creek in the north. And one of the important things to note is that the 2 areas circled there in red are the areas that correspond to the areas of commercial production. And so very important to do your initial seedling screening in the areas where they're going to be grown commercially. What's the result of all that? Well, we compared the 20 best seedlings with the standards that were in the B1.2 trials, and they had similar total kernel recovery and similar canopy volume, but the nut and shell yield was increased in the seedlings. The best 20 seedlings on average had a 39% increase. So that's a big increase in one generation. That's based on single seedling data, and so it needs to be validated with, in clonal trials in the regional variety trials. When you're trying to figure out how many seedlings you need to grow, one of the things that's great about a quantitative breeding program like we've got is there's all this data that you can mine to get information about what you should do in the future. And that's what we've done here. This is looking at the different traits that are of interest to us and working out a threshold above which we need the seedlings to be, and then what percentage of the population in the past have had that level. So working on 2.4 tonnes of kernel per hectare, the industry standard is 0.8 tonnes per hectare. So that's 3 times the industry standard. So it's a really high bar. If you have 38% kernel recovery, that means that 38% of the weight of a nut and shell is kernel. Then that corresponds, the 2.4 tonnes corresponds to 15.2 kilograms per tree. And when we looked at our populations, we found that 0.58 of 1% had above that level of nut and shell yield. And then we've done a similar sort of calculation for all the other characters that we're interested in. And what it works out to is a probability of 0.0016, which means that if we grow out 10,000 seedlings, we would expect to obtain 16 elites. There's a lot of caveats with that sort of calculation, and we're happy to talk about that later, but it gives a rough idea. Those 10,000 seedlings, by the way, that would take about 33 hectares of trial land, so it's a big area to plant out. But that's what we're aiming to do in this next 5-year project. I'm going to talk a little bit about rootstocks and the different species that are involved with macadamia. I passed around those 2 bags of nuts. There are 4 species of macadamia, and the ones in red, that's good, I can hear them jiggling, so that means they're passing around. That's great. The ones in red are the tetraphylla, and the area in green is indicating where the integrifolia are. The blue and this is the little nut that's being passed around. That's the ternifolia, and there's one tiny little yellow dot just up here, and that's the Jansseni. And Jansseni is a bit like the Wollemi pine of Macadamia in the sense that it wasn't known up until two decades ago, about 20 years ago. It's a newly named species. It's only known in one small location. There's probably only about 30 plants in total that are known of it to exist. And that's the one that's indicated in yellow. Now, this graph that's on the right, the open circles represent the cultivars, and the coloured circles represent the wild species, and the open circles are the cultivars. So you can see that most of the cultivars, most of the open circles, are integrifolia. There are a couple here that are hybrids between integrifolia and tetraphylla, and there's one out here that's a hybrid that has some ternifolia in it. And we've just completed some rootstock trials last year. These were trials that were set up by CSIRO, and we found in those trials that rootstock accounted for about 19% of the variation in yield, whereas scion accounted for 72%. There were a couple of age interactions that when we adjusted for those, it could increase the rootstock component accounting for yield variation up to 42%. So what that's telling us is that rootstock's accounting for between 19 and 42% of the yield in macadamia, which makes us think that we're still on the right track in concentrating our efforts on scion breeding. But it does show that rootstock is an important component and something that's worth having a look at. And so we've started doing some work in Jim Hannon and John Wilkie's small tree high productivity project. And what we've done is looked at all 4 species of macadamia as potential rootstocks and a couple of related genera that used to be called macadamia. This used to be called Macadamia floidea, and this used to be called Macadamia grandis. But the splitters got into that rather than the lumpers, I guess, and they've put them into different groups. So the very first thing we're doing in the rootstock screening is to see how well they will produce from cuttings, rooted cuttings. And what we found is that by far the best was the Jansseni in terms of ability to root cuttings. And of the macadamia species, the ternifolia was the worst. So just to summarise about the macadamia breeding, it's been going for 23 years, started by CSIRO, only 2 generations, working with material that was only 2 generations from the wild. The first cultivar release is due to be in 2017, and the best seedlings from the material that's been produced have yield increases greater than 30%. We're looking at alternative breeding strategies, and we started doing some screening work on rootstocks. I'm going to switch tack at the moment and talk a little bit about the peach breeding. I'll give you some background on the breeding, talk about the elite selections that are coming out of the program, and give you an example of how we use imported germplasm. It's really interesting to contrast the difference between macadamia breeding and peach breeding. They're so different in so many ways. And one of the ways is that macadamia breeding, we have all the germplasm right on our doorstep. And in fact, we've got 2 ex situ germplasm collections, one down at Alstonville in New South Wales and one at Tyro in Queensland, where we have about 100 accessions that have been collected from the wild. And that's a great resource to have for breeding project and for allied research. In contrast, peach, the centre of origin for that is China, and we have to scour all around the world in order to get new germplasm. Fruit bats are a huge problem for peach growers and for our breeding project at Nambour. And fruit fly control has always been a big problem, which we've had the luxury of using fenthion, labacid, for control right up until this year. As of this year, it's been banned, and so growers had to go to a totally different control system for fruit fly control using baiting techniques. But another possibility is using netting, and we've used all fruit growers in low chill areas use netting to protect from birds and bats, but we're also looking at using netting to actually exclude fruit flies, very fine netting. And that's going to be a real bonus for we're getting some new netting established at Nambour this year, hopefully, and that'll be a real bonus to our breeding project. The subtropical or low chill peach production is in these major areas, mostly in the same area as Macadamia, northern New South Wales, southern Queensland, a little bit up on the Atherton Tablelands, a little bit in the Hills district of Sydney, and some over in WA. With the change in the fruit fly regulations, high fruit fly pressure areas, like on the coastal areas, it'll be much harder to grow peaches there. And so I'd predict that we're going to see expansion further inland around the Kingaroy region. And I think we'll also probably see expansion in some of the temperate areas who have very early production but can escape frost damage to the low chill flowers, low chill peach flowers. I keep talking about low chill, and I should explain that. My brother, Andrew, I told him I was going down to Bangalore to have a meeting with some low chill growers, some low chill peach growers, and he said, "Oh, what does that mean? Does that mean they're just particularly relaxed growers?" I said, "Well, some of them are, but no, that's not what it is. What it's about is that peaches are a temperate crop, a deciduous temperate crop, and they're adapted to areas that have winters where there is accumulated 800 to 1,000 hours below 7 degrees Celsius. So that's 800 to 1,000 chill units in simple terms. If we want low chill peaches, peaches that can grow in the subtropics, they will only get from 100 to 400 chill units each winter in order to break dormancy. And so that's one of the main things we're breeding for is adaptation to a subtropical environment where the peaches don't need much chilling during winter, and so they're low chill peaches. The other thing that we're breeding for is more compact flowering and ripening. The flowering and ripening extend over 6 to 8 weeks for a single tree, and so that makes it very expensive and very difficult to agronomically for the growers. And one of the big things with any of the soft fruits is fruit quality, size, firmness, flavour, colour. We're aiming for that to improve all those things. In contrast to the macadamia, again, the trees we can grow at very high density. We've planted over 30,000 seedlings and evaluated those, and this is the sort of density that we grow them at, 4 metres between the rows and double beds with 30 centimetres between the trees. And we've released, for large-scale tests, 2 peaches. They're in stage 1 PBR, plant breeders' rights, and also a nectarine, this one, Q3259, that's going through PBR at the moment. And we've appointed a commercialising agent to commercialise some novel selections, things that have either like a flat peach or a flat nectarine, pingtao, Chinese call them, all yellow peaches, or nectarines that are all yellow and then that have got non-melting flesh, and the trademark for those is nectarcot, because they look a bit like an apricot and they're so different to a nectarine. So as I mentioned before, we're using germplasm from all around the world in our breeding program, and I'll just mention, we're also breeding for blood flesh, and we have some selections coming out in that work. We started the work with blood-fleshed peaches, and we've since realised that the little bit of fuzz, the little bit of hair that you get on peach skin with the blood red flesh underneath it makes it look a grey sort of colour, which is really unappealing. And so we've switched our work there to nectarines, looking at producing blood-flesh nectarines. And we can do that pretty easily because the difference between a peach and a nectarine is just one gene, with nectarine being recessive to peach. I'll just give you one example of using germplasm from around the world. We imported seed from Mexico, and this was from Querétaro in Mexico, where because of the elevation, they get about 340 chill units. It's a landrace of peach, probably introduced to Mexico originally by the conquistadors when they invaded, and then over many generations just been propagated by seed. This particular one, Nieve, has no red in any part of the plant. It's a single recessive gene that's a mutation in the anthocyanin pathway that causes that. And we've started characterising this material in order to use it in our program. Thing to note here is that it's slower growing and has more selective shoots, so it branches more. To put that in, what it looks like, shows the Nieve tree on the left compared to the standard Tropic Beauty, and you can see the greater number of selective shoots there, which is not a good characteristic, but the fact that it grows more slowly is useful. In terms of the fruit characteristics, the important things here were the FDP, the fruit development period, is much longer, which is not good. The fruit weight, which is smaller, which is not good. But the thing that's of most interest to us is the high sugar level, 16.5 total soluble solids. And there was a variation with the lowest being at 15% sugar, and there were a couple of selections at the highest of 20% sugar, which is really sweet for a peach. So we're interested in using some of those desirable traits from this material: the high sugar content, firm flesh, and the white rather than cream flesh colour, and no red anywhere in the fruit is also a desirable trait. It's a desirable trait, and I'll explain in a second about that. Undesirable traits: the small fruit, the fuzz, the pointy shape, late ripening, and it's a bit too high chill for what we want, and it's a bit too bushy. So we need to do a bit of work on it. But we're interested in using that white flesh as a signal to consumers that this is something special to eat. And so if you have an all-white peach, it will. We just want to get that associated with a high eating quality, and that's how we're going to use it. We've commenced hybridising it with material to get it into the form we need it, and it'll probably take probably about 3 generations to get that ready, I think. I want to finish off just by talking. I said I was going to use this for my own benefit and to get some ideas across to you about things that areas where we want to get collaboration in and future research projects. So I'll just mention that now. First one I'll mention is the husk spot disease nursery. At Nambour we have a population of about 900 seedlings planted at high density. Remember before I was talking about 300 trees per hectare, and that's at a spacing of about 4 metres between the tree within the row. We've actually got a population at Nambour planted here where it's 1 metre between the row, so really high density. And the reason we planted such a high density trial is we're looking at precocity, we're looking at the genetics of precocity. Obviously that's a really important trait for a long generation length tree like Macadamia. But we'll be finished with that trial when it's 5 years old. That's why we planted at such high density. We knew we'd be finished with it early. And at that stage, Femi and I are going to convert it into a disease nursery, a husk spot disease screening nursery. The high density will be ideal for spreading the disease around. It's a really genetically diverse population, and no one's looked at the genetics of husk spot resistance, and so we're going to do that further in that project, which will be starting up in about the next 18 months. And so that's a good opportunity, I think, for a PhD student. The second point I've got listed there is macadamia self-fertility. Macadamias are self-infertile, and yet they're planted in solid monoclonal blocks. And so it's got to have an impact on yield, and some people have measured what impact it does have on yield. We've identified some material that does have different degrees of self-fertility, and we're interested in progressing that further because we can see that that's can be a major selection trait for us going forward in our breeding, but we need to understand better the inheritance of that trait and also the mechanics of how it works. So that's another PhD student project that's a possibility. The third point is genomic selection, and I mentioned before that we've done a tiny little bit of work with Andres Killian at Dart, and that's looked promising. We have 2 stages of selection. There's 8 years of seedling trial followed by 8 years of grafted trial, tree trial. If we can cut down that 8 years of seedling trial to just a glasshouse test with the genomic selection, it would really speed up the whole program. And the last point I've got mentioned there is the feral peach diversity, and that research is involving, there's peaches growing wild. You may have seen them when you're driving around in the car, and you'll see a flash of pink flower sometimes in the spring months. or if it's low chill material, you'd see it now through July. And there's a good population just at Heifer Creek near Gatton. But that material that's gone feral could represent a totally different source of germplasm for us to use in peach that's right here in our doorstep and perhaps doesn't exist back in China where it originally came from. So we're interested in doing a diversity study on peach. Anecdotally, the story is that some of these old peach feral populations came across with the Chinese gold diggers in the 1850s. They brought lots of things across with them, and one of those things could have been peach seed. So I just want to finish up by acknowledging the project team who I mentioned at the start, the different cooperators, Saro, who conducted the Macadamia breeding project up until 2009, the funding from the different sources, and cooperation of our overseas colleagues in the University of Florida and Dr. Perez in Mexico, and to the low chill growers as well.